in the sheet with the 5 tapes on it.
Yes.
Yes. Yes.
That's essentially the technical device we use to count. And we'll go from here. One of the things I want to point out, you see the ruled paper, the lines on the paper,
Mm-hmm.
that helped quite a bit when there were a lot of crawlers on the tape, because you could count discrete areas. All right, now let's see. Let's see if I can go to the next slide. All right, as you see here, these were the tapes collected on April 24th, 2014. And that was before, it was a learning process before I learned about the ruled paper and the ease of counting. And you can see where I had to draw lines on the tape to make the little discrete areas for counting. And you see all the little dark spots. Those are the things that I was counting, and we'll have some pictures in just a minute to make that a little more clear. But with that first set of tapes, where the star is pointing to, that's the data point on the graph that was generated, the average of those counts on those. And you can see the first generation started maybe toward the end of March and peaked probably middle of April. Can y'all see the pointer?
Yes.
Yes.
And then was dropping off toward the end of April, first to middle of May. But anyway, the pointer, the arrow is pointing to the data derived from those particular tapes. Now we move to way over to June the 26th, and you see a circled area on tape number 3. That's the immatures that were being counted on that tape, and they're easily to identify. You can easily tell them apart from diptera that are stuck to the tape. But you see the blue lines, the vertical lines on the ruled paper. That makes it easier to count as you're moving down the tape. And the data generated from that set of tapes is indicated here on this timeline. Now we move on to July the 3rd. You see what we're seeing on the tapes. All those little dots are immatures, and the data generated from those is here on this particular, indicated here. Let's see, we move to the next set of tapes, and this is where we had a tremendous increase in the number of, I guess, crawler stage, the first end star. You see the circled area on tape number 1. Now we move to a photograph of that area, and you can see where, as you're counting several thousand on a tape.
Oh gosh.
It was, it takes a little bit of time, but you have a counter in one hand, and you're looking through, and you're counting like crazy. And what you may have to do, and I did have to, the area between the 2 ruled lines, I had to go ahead and make additional lines on the tape so I had a smaller area to count, and then I could move to another smaller area and count, as we had this many on the tape.
John,
Go ahead.
did you ever resort to estimates, like counting half the tape and doubling it or anything like that?
Well, to be truthful, when there were the times where we had the most, I took, there's a certain number of sections on a tape between the blue lines, and I picked, say, 6 or 7 of those areas and just kind of randomly picked across the tape, and then used that to get an average, an estimated number for the whole tape.
Mm-hmm.
But that was only for, tapes that were simply covered like this one. Okay, this is July 31st, and that's the July 31st data point that you see where the population really took off. Okay, we move over. Now, This is just an example from October the 1st. You see there's a lot less on these tapes, and you know, the data from that particular date is indicated here on the line. All right. As far as taking the data, the spreadsheet that I sent, I guess, Mike, I sent you a copy of it, and I sent Goo a copy of it, and you know, from there y'all can distribute it out to everybody that might need it. What the spreadsheet, it's in the booklet. You'll have tabs on the bottom of the page. This is the height and stem diameter page. You can see we've already placed our tapes last week, and we have the stem diameter at the point where the sticky trap is placed, and our you know, stem diameter ranges from 5 inches or 5 centimeters all the way up to 6 centimeters for the extra tree we're going to probably treat with imidacloprid. We will have the 3 trees like y'all suggested, and the 5 tapes, you know, per tree. We still have to go back and measure the height. We haven't done that yet, and the boxes that are, or the cells that are marked in red, will be filled in with the height of the tape traps. Okay, you see the bottom tab, height and stem diameter. We'll go to the next tab, and that's sticky trap area. Here you see we've got trap area for tree number 1, tree number 2, tree number 3, and the tree that we will be treating, and that's we're intending to collect those on February the 19th. This next row or next line of little charts is for the next collection date, and then the next is for the next collection date, and you will proceed to just copy and paste and go right on down the line. And this is where we'll, you know, after we pull the tapes from the trees, we take a pocket knife and slit the tape and unpeel it from the tree and place it on a sheet of notebook paper, rules notebook paper. And then we'll measure the length of each of those tape traps. They're all 3/4 of an inch wide, and then this automatically is just length times width, gives you the area figure in the boxes or cells that are blue colored, light blue. You'll take the area of the total tape trap, these 5 cells, and we'll copy them to the next sheet, crawler raw data sheet, and paste it right into where I've got 1, 2, 3, 4, and 5 as a dummy number. The actual lengths will go there. We'll take each individual tape trap and we'll count the immatures on that tape trap and enter the numbers, you know, for tape 1, 2, 3, 4, and 5. And it'll give us a total, and then we'll have an average. We'll take these numbers, we can copy them right down to this table, paste them in, and your total and average should be the same, and that'll be a quick check to look at that and that to see that you know, you have everything copied all right. In this particular chart for our table for tree number 1, you're going to have the area, the number on that tape, and then it will calculate out the number per square inch of you know, on those sticky trap tapes.
Hey John, you know you can do that automatically. There's a reference that you can refer to another worksheet within your spreadsheet.
Yeah, I know. But it was hard. The formulas get pretty tricky, and it's easy to make a mistake. This way I found I don't really make too many mistakes, and you know, I don't have to go back and correct things. Anyway, as we're going down, you see you'll have data for tree number 1. This will also be repeated in this spreadsheet for tree number 2 and tree number 3. And then you can take these figures, these average figures in the yellow boxes, and we'll move those down here, and it'll calculate an average for tree number 1, 2, and 3. And the yellow box is going to be the number of crepe myrtle bark scale immatures per sticky tape trap, whereas the number in blue will be the corresponding number of immatures per square inch of sticky trap. We take these figures and we go to our next page in the spreadsheet, and here you'll see average number per sticky trap tape for a discrete date. Here will be 2/19/2015. We'll enter that number for tree number 1, 2, 3, and then the average number, and then for the treated tree that we're going to have, we'll enter a number. We'll have that data throughout the year as we move, and you'll have to change these dates as you go. The yellow boxes indicate the number per sticky tape trap, and the blue boxes will be the numbers for the immatures per square inch of tape sticky trap. On this same sheet, you'll move to the right, and the graphs will begin to automatically be populated. And as soon as you start entering the data, these points will spread out to what they, whatever they are. And as you go through the year, you'll generate the curves, throughout the time interval. And this particular one will generate for the tree number 1, 2, and 3, and then the average. You'll get 4 lines on this particular graph. Now for the immatures per square inch, it's going to do the same thing. We're graphing the this data up here in the blue boxes, and then if you were to graph or were to take data on a treated tree, which we'll do. Our treated tree is across a sidewalk from the untreated tree number 3, and we'll just graph 3 and untreated number 3 and the treated tree, and we'll have 2 lines on this. The blue line for the untreated will, be exactly like it was on the previous charts, but hopefully the tree with the midichloprid will, flatten out and zero out for a period of time. That hopefully is not too confusing for everybody.
Hey, John, I'm just curious. Last year, did you take crawler data off your treated tree?
One more time, ask. Did I take?
Did you take data off the treated trees last year?
Yes, I did. We had a tree that was treated with dinotefuran, Xylam, and it was, I guess I've shown some of that somewhere, but anyway, it bottomed, zeroed out the population pretty much. And
so you saw the effect on the crawlers?
Yes.
I'm wondering, what do you think about that as a way to evaluate treatments? I mean, we've gone through great gymnastics to count scale, but this seems like a much easier way to do it.
I think this is probably much easier and more accurate way. I mean, more quantified.
This really doesn't take much time at all in the field. You bring it back, and you don't sweat when you're counting these.
That's the best part. And John, can you go back to
one more thing? Let me point out one more thing. Let's see. I'm going to back up to one of those pictures. And y'all were interested in saving your tapes over time. I'll get there in a second. This picture, that's the tape from July 31st last year. I took that photograph this morning. So you see, you can still see after a period of time, and all this tape was just on a piece of notebook paper, and the papers were stacked on top of each other, and with them not being permanent tape, you can actually peel the pages apart and still see what your data or see what the actual counts were.
I've been also using if you use just some clear plastic baggies, and you kind of cut off 3 of the edges so you can open it up all the way and flap it open. And I put the sticky tape, put the first one very near the top horizontally, the next one under it, and so on and so forth, and close the bag on it. And that way you have a double-sided clear baggie, and it's really easy to kind of muck around with them, I guess.
Yeah, you don't have the advantage of the lines being there, though, Irfan.
That's true. I mean, you could put a lined piece of paper under it as you're counting it, I guess. But
I didn't have any problem, not keeping a plastic sheet over them because I kept a piece of wax paper over the top of the stack. But, every week I just took the new pages with the tapes and laid them right on top of the previous week's pages, and then kept a piece of wax paper over the top to keep the dust off. And as you can see, I mean, I took that picture this morning, and the traps were collected last July. And so you can still see, you could actually still count that.
John, here's a dumb question. There's one side with the crawlers, the other side without the crawlers. So
do what now?
For the double-sided sticky tape, there's one side with the crawlers, the other side without the crawlers. So do you...
Well, in reality, there may be some crawlers that get under the tape, but since it's relatively clear, you can actually see through the tape.
So it doesn't matter when you stick this thing on the blue line paper, does it matter which side?
I usually stick the side that was taped to the tree is the side that's stuck down on the paper. But these tapes really aren't stuck that hard to the paper. You can, nick a corner and you could peel the whole thing off.
On this particular picture, on the left side, there are 4 clusters.
Are you talking about here?
Yes.
Here,
yes.
Here?
Yes.
Those are probably nymphs from overwintered females, where that's my guess. They're larger. These are much older, and the new ones are probably first stage crawlers that have come out from the the first generation or so. This was, how do we on the thing? Let's see, on the graph, these were actually 3rd generation. The 3rd peak in the
So John, you didn't attempt to count the big ones, right?
I counted if it crawled on the tape and it got stuck, it was counted as an immature.
So the 4 clusters was counted as 4.
Yeah, they're not clusters at all. They're just larger groups.
Right. So okay, so those.
And they're counted as 1, 2, 3, and 4, right along with the others.
So on this you have some red color things, and you also have some like lighter brownish color. What are those brownish color?
They're just different color variations of the same thing as far as I know.
Okay.
Plus, you're looking at a photograph that was taken this morning, and they've aged 3/4 of a year.
Okay.
It could be just slightly different ages, and some of them have started to develop that pink coloration, whatever it is. Others have not.
Yeah, and I think it's real quick, everybody here. What John does is he uses his criterion. If it crawled on there, then that's what he counts, because the kind of the demarcation, once they settle down and start exuding the felt,
they're not going to crawl on the tape.
Yeah.
When I was counting mine last week, I did see some morphological differences between some of those crawlers, but they all seem to be similar size, so I just counted them all as crawlers. I don't know if some...
That's why, you talked about the first stage being a crawler and then later stage nymphs. Some of those later stage nymphs, they still crawl around. They got legs and they crawl. Mm-hmm. And if they're able to move, I counted them as an immature.
And so that's why we re-headered our graphs, changed them from crawlers to immatures. Correct, John?
Yeah, originally I was calling them crawlers, but then after hearing Mike talk about later stage nymphs, I said, well, you know, I'm counting everything that moves, and that it's crawlers as well as those later stage nymphs, as long as they're able to move and crawl onto that tape. So then I changed my terminology to immatures instead of calling them crawlers or nymphs or anything like that.
John, so Mike, you might want to, Mike, you might want to, we could put in parentheses after that, if that we continue with that terminology, immatures, and then put in parentheses, crawlers, and then what you're saying morphologically you think you may be seeing, so that the reader knows what groups you're up to. Some stage of, the early stage nymphs, I guess. Is that correct, Mike?
Yeah, I mean, that looks like a pretty big change in size between, that might be a third instar nymph that you're looking at in this picture. But
well, and that's why this group needs to, the 3 people or so that are going to do the counting, everybody just, this is the importance of this call, just everybody agrees this is what we're going to,
yeah, just be consistent.
Right. So just at the end of this call, make sure, so if you want to drop the big ones out, that's okay, just...
if you start dropping the big ones out, though, it's a judgment call then on how big to drop out.
Okay.
Well, you can do it by size. If you do it by size, then that's probably as objective as it could be. But what I want to ask John, did those larger ones, were they always just a distinct minority, or did you have some weeks where
earlier in the year, back April time frame, essentially all I was seeing was the larger ones. And then we move over into July, and I started seeing the smaller ones as well.
Now, isn't this picture from a July sample? You got that 0731 number up at the top.
Right, that's the 731, yeah, the end of July.
So you saw a lot more of these early in the season.
Well, let's just look back at another picture. Here you go. That's June the 26th, and you can see,
you think those are the larger ones? Those are the larger ones, right?
Right, and there were no smaller ones on that date on any of the dates.
I kind of think that if there's that much size difference, it shouldn't be that hard to tell those apart. Maybe we should distinguish in our counts between the large and small crawlers.
If you got the time to do that.
What?
If you have the time to do that, it just doubles the...
Well, yeah, I mean, it wouldn't double because I don't know about you guys, but I have a counter that's got, I can count different sizes of things on different buttons, and it totals them up, but I can keep my counts separate. Those aren't that expensive to buy, the little lab counters with multiple buttons. You can also get an app for your cell phone that will do the same thing. You can press different buttons on your cell phone and it will total up your counts, but keep them separate. I'm wondering if we're missing something scientifically important by not distinguishing the larger size from the smaller.
Well, Mike, I mean, even if we don't have to separate the 2 counts, can we count the big ones first, put down the number on the Excel spreadsheet, and then start to count the smaller ones. And like you said, the size here are very different. I mean, John, did you see any nymphs, any immatures that's, the size, the size was, between these 2, it was hard to judge?
Well, yeah. Yes and no. As the generations apparently overlap a little bit, you're going to have all kinds of stuff. You can even, at times you can even see the immatures like this that are even older and have begun to exude some of the little waxy, felty filaments. none of those are pictured here, and I didn't have a picture of that.
But there should be a fairly distinguishable jump in size once
well, the reason I'm hesitating about doing that is you know, I'm the one doing the counting, and when you got it took several hours to count the 5 tapes off of the untreated tree when they were just literally covered with these things. And now this year, there's going to be 3 to 4 times that many to count, and I just don't have all day long to count these things.
Right, I understand.
And make the distinction between I can buzz through and do the best I can, clicking once and counting, but then having to go back and count or make the decision, now is this one bigger than that one, and what am I doing here or there? And it's a time and manpower thing that I don't have the time and the manpower to do it unless Jim wants to count them.
No. By the way, let me throw this out there, just to one possibility, could someone hire a student worker, and anybody that does the collecting sends it to that one student worker that does all of the counting so that there is consistency. And the second thing is I can't, for my drone work, I'm working with a faculty member at Clemson. He hasn't written it yet, but he's got a prototype. But for our aerial digital photographs, he's developing a method where you put the photograph on the screen and then you use your mouse to count, and it makes a mark on the image, and then it counts. And boy, that'd be a huge help, because then, it requires, I think it's linked to a digital photograph, so I don't know if that would help this project or not.
Yeah, Airfon did that last summer with honeydew spots.
Yeah, you can use something like ImageJ as an example of a free software where you can either make it automatically count dark spots on a surface. So if you have a high-resolution image and you're sure that all those dark spots are the scale, you can do that.
Yeah.
One of the things that I would worry about with that, though, at times when we pull the tape off, the little bits of bark, were coming off and staining and whatnot, so that you had a lot of dark places that weren't scale immatures. And
I don't think that's going to work that well here. Yeah.
And can look at that and tell immediately, but I'm not sure that you'd get a...
All right, let's go back to suggestion number 1. Is that a possibility, to hire a student and then everybody send their sheets and have that person, then we don't have these high price counters, and then there's also a chance of variability. Suggestion?
Well, I'm planning to hire someone this summer.
Same.
I'm just thinking of, I want them to do other things too.
Yeah. I mean,
one person will go crazy trying to count all of these.
I'm listening very seriously to you, John. I don't want to make more work than we need to have. I'm just, not having been through the year of counting, I don't really know exactly what to...
When the population peaked in the summer, that was one hellacious time period for counting. I mean, you just sit and you look and you count and you count and you count and you count, and then the next hour you're sitting there counting and counting and counting and counting, and the next hour you're sitting there counting and counting. And that was only on 5 tapes. It's going to be more than that.
Could I suggest that we have a, say, standardized sampling protocol, an estimating protocol? So let's say if it's over, if you estimate over your piece of tape of, however many inches, let's say if it's at least 5 inches long, you got at least 2,000 scales on there. We can say that we will slice it up into, these lines, and you will count maybe 3 random ones or 2 random ones out of that and use that to estimate.
You have discrete areas marked with the blue lines on a ruled piece of paper, so you can pick as many of those as you want and count and estimate then for the total.
Yeah, you could number them and randomly sample them, and say that you're going to sample a third of the tape.
Yeah, because I wonder how valuable it really is when it's numbers that high, the time investment to really know exactly how many they are when you can be just kind of estimating, and it probably is a reasonable estimate.
I think a reasonable estimate is the best way to go when you have too many to count.
Yeah. Do we want to pick a particular, let's say, if it looks like there's over 1,000, let's say, then you only count up to 1,000 of the scales, and then whatever area it is of that 1,000 covered up, use that to scale up, kind of thing?
Well, if you look at the chart here, for most of the year it was less than 1,000.
Perfect.
And it was only from the latter part of July. There was 1, 2, 3, 4, 5, 6 times that the tapes were counted where the number was over 1,000.
Yeah. And would you say then that would work then? Were those the weeks that were the most time-consuming, really?
Oh, yeah.
Yeah.
But the other thing,
it takes no time at all to count down where it's nothing.
Since these preserve so well, it doesn't seem like we'd have to count them all each week. You could backlog them during the worst part of the summer.
Yeah.
In which case, maybe a couple of students, maybe Airfun's going to have a student too. Maybe we could divvy up some of the counting.
So, Mike, are you thinking the estimating is not necessary, or you think it would lose some valuable information there?
No, I think estimating is the way to go, and it's the same. I mean, biologists do it all the time when you're counting E. coli colonies on a petri dish. You count a certain number of squares in the dish, and then you extrapolate what the total number would be.
Yeah.
And I think that's perfectly fair. In fact, we could even make, instead of using standard blue line paper, you could make a paper with a grid on it, and
use graphing paper.
Yeah, that might help, especially for the highest counts.
And then you could really randomly select the squares and count them.
Yeah.
But I think for lower numbers, you just go ahead and count everything. Like, it didn't take me that long to count. I had maybe a couple hundred crawlers on my sample from this week. It didn't take that long to do it. But if it was 5,000, that would have taken a long time.
And I think that's where, I mean, if it looks like it's going to be over 1,000, it's just those few weeks there that they all of a sudden go really high. I think we can create that number. So over 1,000, we all just kind of
Hey,
it's a protocol, part of the protocol.
John, are you getting any money from this grant?
No.
You take a $ and put a 0, and the cost of the tape y'all might have sent. Yeah, that's what I'm getting.
In that case, I don't think you should have to count these at all. I think it should come to Irfan and Mung and me, and we will take care of the counting.
Yeah, that sounds right.
That's quite nice of you.
Yeah, I don't. That's silly for you to have to spend all your time doing something you're not getting anything out of. You're already going and collecting the tapes, and that's plenty.
That's plenty, yeah, for sure.
You guys agree, Mung?
Yes, and I was just going to ask Irfan to get a source, of the grid paper, so, we all get the same paper, and I could order some for John, and then John could put the sticky tape on that grid paper before they mail it to us.
Yeah.
Sounds good.
Yeah, I think that's good. But I do think, when I'm seeing the difference in size between those two nymphs, if they're that easy to tell them apart, I'm thinking we should count them separately.
I agree with that.
Yeah, I think, Mike,
that,
if we're
if we count them as one thing, we do lose some valuable information there.
Yeah. Yeah, and with the estimating, I think, anything above 1,000, it'll make it easier and make it more manageable to count the different sizes.
There's value in counting them that way for, to ascertain the biology of this creature. But my main purpose was to determine the best timing for an insecticide application. And, the difference between the two really didn't make all that much difference to me.
Yeah.
I was looking to see when the best time.
Everybody can appreciate, we didn't know anything last year. I mean, it was just, you know,
Sure.
The first generation, we were so excited. Oh my gosh, we're actually, you know, seeing something, and we got excited at counts of 50, and then it went to 500, and then it goes to 5,000.
The scale was much lower, you know, much lower on the y-axis early in the season, and we got a nice good peak. But as you went further on in the year, that scale expanded up, and your first generation peak just kind of flattened out on you.
Yeah.
John, you applied Xylem dinotefuran at the end of May, I think. So that's you know, that's just somewhere during the second generation. So now with this information,
it was applied. You see where the arrow, my pointer is? In that time, we watched this first generation, and then we saw the uptick of you know, immatures, and we made the application at that point in time, and we were monitoring, and we weren't monitoring that treated tree for the first few weeks after application because we didn't figure it was going to do anything, but we should have to get a baseline of what was there, but we failed to do that. But what we did see when we did start monitoring the population was that it was up about as high as right here, and it dropped at the same time. Essentially, that the second generation dropped on the untreated tree, and then it flatlined for the rest of the season, whereas the untreated tree shot back up. So, I mean, it was doing something.
Now, with the knowledge of the first generation, would you move the application maybe back in March?
Not really, because there wasn't enough to really be, you know, doing anything. There wasn't enough to make sooty mold, you know, make enough honeydew for sooty mold to start at all. And we didn't know what was happening here. When we did make we saw an uptick, so we said, yeah, let's make our application here. Maybe the application should have been delayed until we were at the peak of the second generation. We put it out, it goes up into the tree, and then, you know, you get an effect on this big peak.
Well, there's two other factors that need to be considered. One is that with the dinotefuran, if it appears that it has or is known to have a shorter duration than imidacloprid. And so in the case, the later we went, then the longer, because if you look carefully at the dinotefuran counts, it was breaking loose in the fall. So you could tell it was losing its effectiveness. The second thing is...
But the population line for the dinotefuran treatment was still below that.
Very much so, very much so. But
you did get you were getting some activity on those trees.
Yeah, we were getting renewed activity, whereas I suspect with imidacloprid it would still be near zero. The second thing is there's an aesthetics issue. By the time, and it was just pure luck, but because we treated when we did, it preceded the significant bark exfoliation on the tree type, and so we killed the population, and then as the bark, it sloughed off this really blackened, you know, bark that was there. And so as we've got a wonderful series of photographs that you can see in July or certainly early August, the bark was like nothing had ever happened.
And
the leaves were all green and not covered with sooty mold like the untreated tree.
John, what was the date of application?
It's May the 29th.
I want to say May the 28th.
Yeah, that's what I thought.
So am I correct that prior to that, well, up to the second generation, this graph reflects both the counts from both trees?
No, it's just the untreated tree.
Okay.
It was about June 15th when we started
right along in here.
But they were nearly identical trees. So, you know, we don't have that.
Separated by a sidewalk.
Yeah, they were. So the counts prior to that should be fairly similar to what's on the non-treated tree.
So your title, your caption's a little off. It should be just say immatures on untreated infested crepe myrtle. Right?
Yeah. This was immatures on treated and the title is you know, I didn't edit that properly at the time. It just should be untreated.
This is great stuff, and it's very useful to us. It's going to help a lot with doing it this summer.
Well, hopefully we can duplicate what